Thank you everyone for standing by, and welcome to our webinar entitled A New Modeling Approach for Estimating Stream Phosphorus Cycling from High Frequency Dissolved Phosphorus Monitoring. This webinar series called Freshwater Science will highlight Ohio's Sea Grant and partnering scientists every month. Every quarter is a different focus, from human health and fish farming to harmful algal bloom and human decision-making, bringing applied research to the public on issues that affect our Lake Erie communities. I am Jill Gentis-Benicki from Ohio Sea Grant and Stone Laboratory, and joining me today is Dr. Jim Hood from Ohio State University. Dr. Hood is an associate professor within the Department of Evolution, Ecology, and Organismal Biology, as well as the co-director for the Aquatic Ecology Lab at the Ohio State University. Dr. Hood's research focuses broadly on understanding how global change influences aquatic organisms and the structure and function of aquatic ecosystems, with an overall goal of gaining an understanding of how global change drivers influence freshwater organisms and ecosystems to improve our understanding and management of these resources. We're delighted to have Dr. Hood here today to discuss his research. Before we do that, just a few things about the webinar itself. During our presentation, all participants will be in a listen-only mode. Afterwards, around 12:20, I'll conduct a question and answer session. If you'd like to ask a question during the presentation, please feel free to use that chat feature, and I'll collect all your questions and pose your questions out to Dr. Hood at the end of his presentation. As a reminder, this webinar has auto captioning and it is being recorded to be posted onto our website for later viewing. Also, we will post a webinar survey in the chat feature toward the end of the half an hour. Please take a few minutes after the webinar to fill out that survey. It will help us continue to bring you great webinars. So without any further delay, I'd like to introduce Dr. Hood, who is presenting a new modeling approach for estimating stream phosphorus cycling from high frequency dissolved phosphorus monitoring. All right, Dr. Hood.
Thank you, Jill. I appreciate it. And I appreciate everybody spending their lunch hour with us. And can you hear me well, Jill? They've decided to do a ton of landscaping outside of my window right now.
So yes, I think you sound great. And anyone who has any issues, let me know in the chat feature.
All right, I'll try to speak up. All right, so today what I want to do is basically talk about a lot of the work in my lab focuses on understanding the role of rivers and streams and influencing phosphorus exports. And what I'd like to talk about today is some modeling work we're doing, where we're trying to develop models that'll take advantage of some high frequency dissolved phosphorus monitoring. Before I get started, I'd really like to thank Ohio Sea Grant for funding this work, as well as my collaborators on this project, including Oksana Shurkreti, Emerson Webb, both of whom are in statistics, Andrew Ferris, who was here in EEOB, Joe Gentine, and Devin Maddy, who worked on the project. This wouldn't be possible with all their work, and I'll talk more about their individual contributions as we go. So, harmful algal blooms have been coming up, an increasing concern recently. They seem to be increasing in both the frequency and their magnitude. These blooms degrade water quality. They can be a threat to human health and wildlife and biodiversity. And ultimately, these blooms are driven by nutrient loading from their waterbed. And so a good example of this, of course, is Lake Erie, right? There have been blooms reoccurring in Lake Erie since the mid-2000s. We know that the bloom magnitude in Lake Erie and Western Lake Erie is generally driven by bioavailable phosphorus loading, primarily that's coming from the Maumee Watershed, shown here. And we also know that phosphorus exported to the lake between March and July from the Maumee Watershed is the most important in determining bloom size. So this science has informed these targets, which aim to reduce phosphorus loading by about 40%. And in particular, we want to reduce both the amount of total phosphorus and the amount of soluble reactive phosphorus, or SRP, by about 40%. And we focus a lot on SRP because it's thought to be the most bioavailable to cyanobacteria, and so they can readily take that up and assimilate it. So to achieve these targets, we need to have a good understanding of where the phosphorus sources and sinks within the watershed. Where is the phosphorus coming from? What processes remove phosphorus before it makes it down to the lake? And many phosphorus source attribution studies, whether they're statistical or whether they're modeling, make the simplifying assumption that once the phosphorus leaves the land and enters a ditch, stream, or river, essentially nothing happens to it, and it makes its way down to the river, to the lake. And that's a great simplifying assumption, but it has some implications for how we manage phosphorus. And so why does this matter? This matters if we just think about rivers like they're pipes moving phosphorus from the land to the lake. We don't really need to worry about these streams and rivers. If these rivers and streams and ditches are phosphorus sources, then they're actually part of the problem. And they're a place in the landscape where they're going to require management and restoration. Alternatively, these systems could be a phosphorus sink, reducing the total amount of phosphorus coming into Lake Erie, or at least during times that it really matters. And so in this case, they could actually be helping, and we need to be aware of that and not mess that up. And so I just want to sort of illustrate why this is important to the management of this system with kind of a really simplistic example. So this builds on work that one of my students, Whitney King, did that focused on basically the role of rivers in reducing phosphorus exports to Lake Erie during high flow events. And so during March to June, SRP loads on average recently are 222 megatons. And if the rivers are like pipes, then we can assume that that's 222 megatons of phosphorus is coming off the land, and we need to reduce that by about 88 megatons. But if the rivers are actually increasing SRP by 20%, there's no evidence for that, but this is just sort of a thought experiment, then that means that about 176 megatons is coming off the land. And so we don't need to work as hard to reduce phosphorus exports from the land. In contrast, if the rivers actually reduce SRP, they're helping us mitigate blooms, but we've got more phosphorus coming off the land. So to achieve that 40% reduction, we need to actually reduce it by more. And so, these are really simplistic calculations, but what they illustrate is how important it is to understand what this number is and the role of these river and ditch and stream systems in influencing phosphorus exports. And in particular, we want to know these numbers for time spans that are relevant for managers. So, not one measurement during the summer, but be able to sort of make these estimates over the March to July period or over an annual period. And this has really been a significant scientific challenge for three reasons I want to illustrate. And it's part of the challenge that we're trying to address in this work. So the first issue is that freshwater phosphorus cycles are really complex. So this is a diagram put together by Lauren Kinsman-Kinsella. That's it's a simplification of a phosphorus cycle, but you can see here on the left-hand side, it basically just shows the movement of phosphate, which is dissolved, into particulate fractions. And so on the left-hand side, we have uptake of phosphorus into algae or into plants, and also the remineralization of phosphorus from bacteria that might be breaking down organic matter. So biology on this side, abiotic processes on this left side, and there's a whole host of abiotic processes that could be important, but I could highlight the absorption and desorption of phosphorus onto sediment particles, co-precipitation of calcium and phosphorus, things like that. So we have numerous processes, and so when we go out and we measure this stuff, we have to make guesses about what's important. And that's often based on previous work and also where we have our expertise. The second issue is that these things are very difficult and time-consuming to measure. So often we have to focus on a particular process. Measuring that process might take, a day, might take 2 days if we want to measure two processes. And as a result, the measurements we take tend to be relatively infrequent. Measuring monthly or maybe biweekly is a high-frequency data set for this. And so you can imagine when you're getting these data sets that are monthly, that makes it really difficult to sort of integrate across a 6 month period with any reliability. The third point is that there's high spatial variability in phosphorus cycling in streams. And I could show you a number of great papers that illustrate the spatial variability. But I think if I just tell you that, I've already told you that it's the amount of plants and sediment and algae that's driving this. And these are just, 8 pictures that I put together of different systems that we've worked in. And you can see sort of the diversity and the flow environment and the amount of plants in some systems versus in other systems, the amount of plants versus algae. There's an incredible diversity here that leads to a wide diversity across the Maumee watershed and what streams are doing, whether they're sources of phosphorus or sinks of phosphorus. And then the final point I'd like to make is that, and all this is fairly self-evident, but there's a lot of temporal variability in these processes as well. Temporal variability both between the day when we sample and nighttime when different processes are important, and also, from month to month and across seasons. So for example, we know that, algal growth and phosphorus uptake is controlled by light and temperature, and there's seasonal patterns. And so we might expect algae to be more important during the summer than they are in the winter. Similar to that, we know that on the abiotic side of this, that things like phosphorus absorption, desorption are controlled by processes like temperature and dissolved oxygen and pH, all of which vary over time and are influenced by the biotic side of this. So thus we expect, considerable daily and monthly variation in phosphorus cycling. And so the challenge here is try to be able to integrate across all of these seasons and come up with good estimates of what's happening with phosphorus cycling across this at scales that are really relevant for annual and seasonal budgets, and for understanding what role these rivers are playing between that March and July period. And I often feel very uncomfortable doing that from monthly measurements. So that's part of what really motivated, that's the challenge that motivated this study. And I think this is something that is doable. 2 trends suggest that this is something that might be doable. The first is there's been a great increase in the number of studies that estimate parameters of interest, whether that's like predator-prey dynamics or primary production or methane production from high frequency time series data sets. So these modeling approaches are becoming more usable to normal ecologists like myself. And second, there's been this proliferation of sensors like this hydrocycle phosphate sensor and these new lab sensors kind of across the United States that provide this high frequency. And by that, I mean like sampling every 2 hours phosphate data. And so here's an example. These hydrocycle phosphate sensors were used in these USGS super gauges, all of the purple ones here, and many of these have this hydrocycle phosphate sensor. So the goal here of our project is to develop a modeling approach for estimating aspects of stream phosphorus cycling from these high frequency dissolved phosphorus measurements. And this work really builds on work that was done by Matthew Cohen in this 2013 ecological monographs paper. And they were focused on these springs in Florida. And they used one of these hydrocycle P sensors. And they used the diel or day-to-night variation in dissolved phosphorus concentrations to estimate autotrophic and heterotrophic phosphorus uptake, as well as the precipitation of calcium and phosphate. So this was a great study, and this really sort of excited me and got me thinking, but it has some limitations in that their modeling approach is very system specific, and it required a high degree of user input here. So this sort of stuff informed 4 modeling goals for us. The first is to estimate in-stream dissolved to particulate phosphorus transformations. So what I was showing in that diagram at a sub-daily frequency, so every 2 hours. We wanted to estimate both the net whole stream phosphorus transformation rate, as well as the rate of specific subcomponents of both abiotic and biotic phosphorus uptake and release. We wanted to develop a modeling framework that could be easily generalizable to other streams and rivers. And we wanted to create something that could quantify the uncertainty in our parameter estimates. And so to do this, we collaborated with Laura Johnson at the Heidelberg National Center for Water Quality Research And they have these two sites here, one in Potato Run and one in Shella Run, both in the Blanchard Watershed, where they've installed a new lab, which is measuring total phosphorus, SRP, total nitrogen, and nitrate every 2 hours. And it's next to a USGS gauging station and a number of different sensors. And so as part of this project, we combine this with monthly monitoring for a bunch of aspects of the stream phosphorus cycling occurring, including like benthic and suspended organic matter, whole stream phosphorus uptake rates, sediment phosphorus absorption and desorption, as well as zero equilibrium phosphorus concentration, which I'll explain in a minute. And then we also use dissolved oxygen sensors to estimate primary production and respiration for the stream. And I'll note here that this project started in February 2020, about 2 months before COVID. And so this is like the typical COVID project, right? Project was initially stopped, then we struggled to get it going. We had one student working on this project who decided to go in a different direction with their life, 2 technicians working on this project who decided to go a different direction with their life. There was issues with the new lab. So a lot of what you're hearing here is the result of adaptations to these dynamic conditions. So with that said, let me talk about our model here, and then I'll show you some results. So here's our overarching model, and you might see this and think calculus, but it's not that complicated, right? All we're trying to do is think about the change in SRP per unit time, and we're arguing that it's going to change with 3 processes: autotrophic uptake, so how much phosphorus algae take up; heterotrophic uptake, so how much phosphorus bacteria mostly, and maybe fungi take up. And we recognize that heterotrophic uptake can be bidirectional, so it could be positive or negative. And then autotrophic or abiotic uptake, which sort of encompasses all of these processes. So for autotrophic uptake, we basically argue that we can model this as a function of primary production and primary producer phosphorus to carbon ratio. So if we know their P to C ratio, we can estimate how much phosphorus they take up. This is basically driven by our metabolism data. So we take that information and we estimate P to C ratio. For the heterotrophic uptake, so this is like bacterial phosphorus uptake or release, that's driven by heterotrophic respiration and this heterotrophic P to C ratio. And then finally, for abiotic uptake, we use a relatively simple formula here, which has 2 components that matter: this daily maximum uptake velocity. And so this is essentially the rate at which dissolved phosphorus moves into a sediment particle or moves out of a sediment particle. And it's the maximum rate that can occur at. And then we multiply that by the SRP concentration of the stream minus this zero equilibrium phosphorus concentration. And so EPC 0 is essentially the stream water concentration at which sediment doesn't release or take up phosphorus. And so if we look at the relationship between EPC and stream water SRP concentration, if those are equivalent, the sediment isn't releasing or taking up phosphorus. If EPC is higher than the stream water SRP concentration, the sediment releases phosphorus. And if it's lower, then the sediment takes up phosphorus. So this is a relatively simplistic model that can be applied in a lot of areas. And then Emerson and Oksana and I work together to sort of, this is really like Emerson and Oksana's work, to develop a way to model these parameters. So they use a Bayesian approach to estimate parameter values. And we focused on estimating 4 parameters: autotrophic P to C, heterotrophic P to C, this maximum daily uptake velocity, equilibrium phosphorus concentration. For those of you who care about this stuff, I can talk about it more, but briefly, we use partial pooling to estimate V sub F daily. We modeled both the process and the observation errors. This was implemented in STAN using RSTAN and R. And Emerson did this by running 4 chains with 20,000 burn-in and 20,000 sampling per chain. So I'll talk about the results of this in a second. I just want to start by showing you some of the monitoring data that we have. And so this shows SRP concentration from the new lab from about May through mid-September. You can, and then the red is discharge in the stream. I forgot to mention we're only focusing on one stream here for this talk. And I'll note two things here. Whenever discharge goes up, SRP concentration goes up, not that surprising. And we have this period here where we lost a bunch of data because there was an issue with the new lab. So for the purposes of this modeling project, we're just focusing on this 10-day period during relatively low, when base flow was relatively stable. And we did that to basically test and develop this model with the hope that later on we can further implement it under tougher conditions. So you can see under this period, SRP concentration is decreasing. There's a lot of variability, and discharge is also decreasing during this period. This plot shows our gross primary production data from metabolism calculations. And so this is SRP concentration here in the black dots. The light just highlights the light during the day. And then these are our GPP estimates every 2 hours. And you can see that they kind of increase over time, after fall over this period, and then they stabilize. And what I want to highlight here is that it looks like SRP is really variable. But actually, if you look, there are these nighttime peaks in SRP, basically after day 4, which suggests that there's some biological process, likely GPP, that's driving down phosphorus concentrations during the day. And then I want to make one final point about these streams. This plot shows our equilibrium phosphorus concentration and SRP data. And so equilibrium phosphorus concentration is in the blue here, it's very low. SRP is in the red. SRP is higher than EPC, so that means that the sediment in the stream is likely absorbing or binding phosphorus, except for on this one date here where it seems to be releasing phosphorus. And this is consistent with what we see throughout a lot of streams. There's temporal variability in whether they release or bind phosphorus. All right, so with sort of that background led, how did our model do? Our model did a reasonably good job predicting these dynamics. So this is SRP data, a new lab SRP data in the gray dots. And then the blue is the mean posterior predicted estimate from the model. And then the ribbons illustrate the 95% credibility intervals. So you can see that the model's capturing this downward decline in SRP concentration, and it's also capturing some of this diel variability as well. I'm just going to give one example of these sort of parameter estimates, but there was good correspondence between our modeled and our measured equilibrium phosphorus concentration data. So this shows EPC here that we measured in the field over that year, what I showed you earlier. And this is the model estimate over that 10-day period. And the model is a little bit higher than what we saw in the field, but not by much, but there's fairly wide error bars around that. And in both cases, these indicate that the sediment is a sink for phosphorus. So we can take this information and estimate the contribution of each of these compartments, the autotrophic, the plants and algae, the heterotrophic, the bacteria, and the abiotic compartments to phosphorus uptake or release in the stream. And that's what's shown here. So this is the posterior mean phosphorus uptake rate for the stream. I'm just going to show you the first day so you can get your bearings. So red is abiotic uptake. It was the highest. Green is autotrophic uptake, second highest. Blue is heterotrophic uptake. And so here's the 0 line. So the heterotrophs are actually releasing phosphorus, whereas the abiotic processes, according to the model, and the autotrophs, according to the model, are taking up phosphorus from the water column. And so if we look at this whole 10-day period, we see that this pretty much plays out. The autotrophic uptake increases with GPP, which isn't surprising. There's abiotic goes up for a while and then comes back down. And the heterotrophic uptake stays sort of negative throughout this period. So this was really exciting to us. It seems as though we can estimate these processes, and then what we can do is sum all those together and estimate on net what is the stream doing over this period. And that's what's shown here. So that's the sum of those three compartments. And we can see that and here's 0 for reference, and here's the estimate, the mean estimate, and the 95 credibility intervals. So on that first day, it's relatively high, comes down, and it's stable for this period. And then it goes down to about a net of 0 on this last day of measurement. Suggesting that, so together this suggests that over this period, the stream is a net sink for phosphorus. And then we're still working up our whole stream uptake data, but we can compare this to some measurements that Bernot made in Indiana and Michigan ag streams. So this shows whole stream SRP uptake versus SRP concentration. You can see our estimates here are a bit higher than theirs. Theirs ends at 0.86. That'd be right here. But our SRP concentrations are also a bit higher, and they predict this positive line. So this suggests that these model estimates are in the ballpark for these estimates. So just to conclude, I would say our model performed well for this very limited summertime period we examined in this test case, although our model has some limitations, which I'd be happy to discuss. Our model indicated that over this limited period, the stream was a net phosphorus sink, but the heterotrophic phosphorus remineralization was pretty considerable. It suggested that autotrophic and abiotic phosphorus uptake was of similar magnitude. It was in the same ballpark. And the model provided reasonable estimates of our zero equilibrium phosphorus concentration when we used a prior that was sort of informed by field data across a wide number of projects. So taken together, this model suggests that this model could be potentially useful for estimating phosphorus cycling streams over longer time periods. But there are some limitations that may need to be evaluated, which I didn't talk about in detail here, but they include some identifiability issues in estimating the parameters, sort of some uncertainty for myself about how this deals with rapid changes in discharge and SRP concentration. And then there's also the issue of the cost and the reliability of those sensors. So with that, I'd like to thank you for listening, and I'd be happy to take any questions if that's all right, Jill.
All right, well, we have gotten some really great questions during Dr. Hood's presentation. So let me get started and ask as many as we can here. First question that I have is, and this was, I think, early portion of your presentation. Were you able to estimate the amount of sediment phosphorus that was used by macrophytes and released as DRP to the stream?
Like this, so maybe the amount of sediment that was trapped by macro. That that is not a specific process that we so with a modeling approach, it may have captured that in one of those two compartments, either in the abiotic compartment, likely in the abiotic compartment if it's what the sediment is doing. We took some sediment cores and measured the potential for phosphorus release from sediments, but we did not, and we also took sediment trap data, but we did not specifically look at how much sediment is being trapped by the plants. But under some conditions, plants can be incredibly important for both trapping sediment and phosphorus.
Okay. If the sediments are generally a phosphorus sink, then the sediment load to Lake Erie should be considered part of the phosphorus load, shouldn't it?
Well, okay. So thank you for that question. I mean, I would make 2 points. So in this stream, it seems like based on our measurements that the sediment is generally a phosphorus sink. In other streams within the Maumee watershed, it's not. Sediment is a phosphorus source. So it's important to remember that there's a lot of variability across the watershed, particularly during low flow periods. And then the second part of that question is, so should that sediment be considered part of the load? It's only considered part of the load once it's exported from the watershed. But it definitely, if we need to, I think we're kind of coming at the same point from different directions. If we want to understand what's coming off the land, then we need to sort of understand what's being trapped in the ditches and rivers, and may it stay there for months, years, decades, and then eventually be exported.
All right. We got a couple of questions dealing with this modeling approach of whether or not could this modeling approach be applied to like extreme events like flooding events?
My guess is no. I think it's just things are changing too quickly, and one of the things that we need to evaluate is at what point does the model break down? But I think under extreme events, it's just discharge and SRP is changing too much that we can't get a good signal with 2-hour data.
Yeah. All right. Another question I had, with this model being site specific, would you be able to comment on net transport of phosphorus downstream?
Well, you could use the underlying data to, well, okay, what do we mean by net transport? I don't know. That's sorry, that's a really good engaging question, but I would have to. I think with a little math you could estimate. I mean, obviously we're estimating how much phosphorus is taken up over a certain period of time by the stream, and we could use the same data to estimate how much dissolved phosphorus is being exported. So we could come up with a net estimate. Sorry, I had to work through that.
Could you talk a little bit, does water temperature impact the total phosphorus, dissolved phosphorus, transformation processes?
That's a good question. Water temperature influences not all, but many of the underlying aspects of phosphorus cycling. And so, algal uptake is influenced, and algal growth is influenced by temperature. Many of the abiotic processes are influenced by temperature. We didn't explicitly incorporate it into this model, but one could.
We got a couple of questions of whether or not this model could be used to improve models like SWOT.
I don't think so. I think they're coming at this model. I think what this model is really useful for is estimating these parameters. How much phosphorus is net phosphorus uptake over a day, over a period. SWOT is trying to predict that based on other things, but it could also could help provide some estimates for SWOT. That's one of the problems. SWOT with a lot of the in-stream parameters, you kind of have to do an informed guess with what those parameters are, and this could help inform some of those, I think.
Okay. I have two more questions because we're at 34, 35, so I don't want to keep everyone too much longer. But I appreciate the fact you're willing to answer all these questions. What explains the inverse relationship between heterotrophic and autotrophic release / uptake?
I think it's a good question. I've thought about that. So have the rest of us working on this project. So autotrophs, during this period, you saw kind of gross primary production going up, right? And so autotrophs are growing. They're taking up phosphorus to build new tissue and stuff like that. So it makes sense that they're sort of a net sink for phosphorus over this period. The heterotrophs, they could be growing, or they could be, I don't know what the stream was like at this period, unfortunately. But if there's a lot of, algal mats that have bloomed and started breaking down, which happens over and over on summer, especially kind of right after these storm events, it could be that the bacteria in the stream are breaking down these algal mats, and they're just essentially releasing the phosphorus that was in the algal mat. So there's this loop between the autotrophs, phosphorus going into the autotrophs growing, dying, and then the heterotrophs releasing it.
Okay. I have a what if scenario for you. Are you ready? Okay. If a shallow channel passing through a cultivated crop field was replaced by a subsurface drain tile, would you expect the total phosphorus and what is SRP? SRP in the water leaving the crop field to be lower? In other words, would surface water runoff convey more total phosphorus, SRP, than water forced to percolate through the soil profile and exit as a drain tile effluent?
That's a really good question. I just don't know.
Okay. I wasn't sure about this what if, so I thought I'd throw it out. So that's totally fine.
Yeah, I'm gonna be thinking about that for the next day.
Okay. One last question. Well, actually two. I had one person that was very curious about what's happening with nitrogen, that you mentioned collecting data on nitrogen. So do you have any insight on that?
No, we don't. I mean, that's one of the problems here is there's only, as researchers, we only have so much bandwidth we can handle. And, even the monitoring that we were doing in 2 streams focused only on phosphorus, only on some subset of phosphorus. That took basically a week of sampling a month just to get the samples and process the experiments. So unfortunately, even though I totally agree that nitrogen is important, I would love to know that, it's just not something we've been able to do.
All right. One last question. People were asking whether or not this data will be leveraged by H2 Ohio and NPS projects at watersheds downstream.
I think that's a question you'd have to direct to Laura Johnson.
Okay. All right. And then the only other thing was we had several people asking if they would be able to contact you.
Yeah,
is that okay? And what is your email address?
hood.211@osu.edu.
All right, 211@osu.edu. I will put that in the email that goes out to everyone so that they all know. All right, well, I think that was the bulk of the questions that we had gotten, and thank you for staying with us a little longer than the half an hour. So I just wanted to close out this webinar and thank again, Dr. Hood, for your willingness to talk with us again today on your research. This was really an excellent discussion. I also wanted to remind everyone that the survey URL for this webinar is in the chat feature, so please take a few minutes to fill that out. This webinar series is sponsored by Ohio Sea Grant and will continue on in September, September 13th with Bowling Green State University's Vern Bingman about his research on bird migration and the Lake Erie Islands. The registration link is in the chat. Again, thank you, Dr. Hood, and all the participants on this webinar. We hope this was beneficial and hope you'll join us on into another upcoming webinar. Thanks again, Dr. Hood. This was really fantastic. We really appreciate your time.
Thanks, Jill. Thanks, everybody, for listening.
Thanks, everyone. Have a great afternoon. Thanks again. We appreciate it.
Appreciate it. Take care. Have a good day.
Bye.